Great, thank you. I'm going to have Nick drive the slides for us today. So thanks very much. And I'd like to share with you the ongoing work in the Adaptive Oncology Project Heterogeneity in Time that looks at the sequential profiling of samples, whether they're tissue or liquid biopsies that have been obtained from patients in clinical trials to identify biomarkers of response or resistance to treatment. So just a little bit of the background to the project. One of the themes of the Adaptive Oncology Project in the last strategic plan was to develop tools to measure heterogeneity in two broad ways and to use these outputs in clinically meaningful ways. So first, in the context of spatial orientation of cancer foci between one another, but also temporally in the context of changes under the selective pressure of treatment. So with a measure of heterogeneity, we can then begin to look at the impact as it correlates to clinical variables, including outcome. And this allows clinicians to better prognose, but also to predict response to treatment. So as you can see, there are challenges that are posed to successful treatment of adaptive changes which occur within tumors over time, and we need to exploit these changes for the patient benefit. So the Heterogeneity in Time project will perform deep molecular profiling of sequential samples from breast cancer clinical trials to develop those molecular tools for assessment of adaptive changes over the course of treatment, to identify key drivers of drug resistance across the treatment spectrum for breast cancer, and then importantly, to disseminate that knowledge provincially and nationally, and of course globally, and to develop contemporary adaptive clinical trials. So next slide, Nick, please. Thank you. Just a little bit of background to breast cancer and the clinical need. 1 in 8 women will develop breast cancer in their lifetime, and more rarely, 1 in 800 men will develop breast cancer. Approximately 15% will die from their disease. And hormone receptor positive cancers are the most commonly seen cancers in the clinic, approximately 80% that are seen. Despite their initial responses to anti-endocrine therapy, of those, another 50% will experience a recurrence. And as I mentioned, they do first-line treatment is anti-hormone therapy, but depending on the presentation, patients may also receive a chemotherapy or radiotherapy. At recurrence or in the later metastatic setting, there are a number of targeted therapies that are being used, including CDK4/6 inhibitors, mTOR kinase inhibitors, and PI3 kinase inhibitors. And what we need now are to identify or better stratify those patients to these targeted therapies, because there are no solid biomarkers that will help us to stratify or to predict their response to these targeted therapies. So to improve the clinical management of breast cancers in both the early and the late setting, we need more robust biomarkers for response to treatment. We also need to leverage the ability to detect these biomarkers during the patient's journey, a cancer journey and their treatment, and this can be done through the profiling of liquid biopsies. Next slide, please, Nick. So the background to MA38. MA38 is a randomized trial for women who have hormone receptor positive, HER2 negative, advanced or metastatic breast cancer. They're randomized to 2 different dosing schedules of the CDK4/6 inhibitor palbociclib, as well as anti-endocrine therapy. Biopsy tissues, pre-treatment, and 3-month interval blood samples to the point of progression or to the end of the study were collected and banked. And to the right, you see the mode of action of the CDK4/6 inhibitor, palbociclib, together with the anti-hormone pathway signaling, helps to inhibit cell cycle progression primarily through RB. Next slide, please. So to identify markers of CDK4/6 resistance, the project involved the targeted sequencing and transcriptional profiling of patient biopsy at diagnosis, as well as the targeted sequencing of the ctDNAs extracted from the pretreatment, progression, and intervening time points. For the tissue profiling, we use the MSK impact panel of pan-cancer prognostic and predictive genes in collaboration with George Reyes-Vito. Transcriptional profiling was performed by Diagnostic Development using the NanoString Breast Cancer 360 panel of 770 genes. And these are genes that are also part of signatures that are relevant to the biology of all breast cancers. Profiling of the ctDNA was performed using a custom design panel and processed through Trevor Pugh's team at TGL. And very shortly, I'm going to hand it over to Nick Cheung to present some of the preliminary analyses on the data so far. And this will be covering the tissue profiling and the ctDNA profiling of the baseline samples. We still have about 600 more ctDNA samples that span this period of time, and they're currently being processed. So it's still very early days, and we'll be seeing a lot of amazing data coming out of this project very shortly. So I'll hand it over to you, Nick.
Thank you, Jane. Hey, everyone. So as Jane mentioned, we do want to address several aims with regards to some of the data that we've been given. So as Jane mentioned, we're looking at data from 3 different sources. RNA expression from the solid tumor biopsy, DNA mutation sequencing of the tumor biopsy as well as mutation profiling of the plasma cell-free DNA. So here we want to identify different subtypes of breast cancer that are either susceptible to treatments or identify biomarkers that confer resistance to palbociclib. So breast cancer itself is quite heterogeneous at both the morphological and molecular levels. In the clinic, breast cancers are typically subtyped based on the presence or absence of hormone receptors, estrogen or progesterone receptors, as well as whether the human epidermal growth factor gene HER2 is amplified or overexpressed. And these are typically subtyped using immunohistochemical assays, and it's primarily the main molecular assay that is being used at the moment for patient treatment decision-making in the clinic. However, even among these broad clinical subtypes, breast cancers can typically be stratified even further into intrinsic molecular subtypes, according to the expression profiles of 50 different genes. And these subtypes are luminal A, luminal B, HER2-enriched, basal, and normal-like. So as you can see here on both plots, so on the plot here on the right, we've essentially conducted a PCA and have labeled each of these samples by their tumor subtypes. So as you can see, gene expression does seem to resolve some of the intrinsic subtypes within our samples. But in addition to that, we've also profiled the mutations in the solid tumor as well as plasma cell-free DNA. And what this plot is showing you is the overlapping samples we have at the moment that have both solid tumor mutation profiling as well as mutation profiling in cell-free DNA. So far we have about 48 samples that have both assays conducted, as well as most of these also have had their gene expression or transcriptome profiled. And as you can see, among those that have been profiled, around half of them are luminal A and half of them are luminal B. So in addition to that, we at the start, what we try to do is we try to look at the variants that are being captured in plasma cell-free DNA versus those that are seen in solid tissues biopsies. So for those that are unfamiliar, cell-free DNA is DNA that sheds freely in the blood, and it's primarily shed from bodily tissues, including blood as well as tumors. And cell-free DNA itself as a biomarker in recent years has garnered a lot of attention as a potential non-invasive biomarker that is reflective of the tumor's genome and can potentially capture both genetic and epigenetic alterations in the blood that is concurrent with what is seen in the originating tumor. And there's been an increasing number of assays using cell-free DNA mutation profiles to assess different things like treatment efficacy, assessing minimal residual disease and recurrence. But there are often issues with using cell-free DNA in distinguishing somatic variants versus those that are germline, or identifying somatic variants that is specific to the tumor versus those from other tissues such as blood. So what you notice here is that we do capture slightly more variants in the plasma cell-free DNA versus tumor DNA. And if we look at the breakdown of the classes of variants that are being captured between the two, we see that the largest difference is when looking at these missense mutations. So these essentially confer a change in amino acid base. And we suspect that a lot of these mutations that are being captured in plasma cell-free DNA might not be concurrent with the tumor. It could be somatic mutations that are seen in blood cells, such as those from age-related clonal hematopoiesis. And the benefit of this study here is that not only do we have the solid tissue tumor biopsy, but we also have repeated sampling of the plasma cell-free DNA. So identifying what is tumor specific is something that we're aiming to clarify as well without a matched normal sample. And just to give everyone an overview of sort of the variants that we're currently capturing across the plasma as well as the tumor biopsies. So here on the left is a waterfall plot, and each column indicates a solid tissue biopsy sample. And each row is showing you the genes that are being mutated in each of these samples. So as you can see, we do capture a lot of the canonical drivers. Most of the variants that were mutated in the solid tissue biopsies were PLK3CA and TP53. But as you can see, there are quite a few other there are other driver mutations that are also recurrently mutated in more than one sample. We also try to see if we could just use these mutations to resolve some of the different intrinsic subtypes. So what we did is we used these mutations as well as their VAFs just to see if we can cluster them and label them by the different subtypes. As you can see, compared to using gene expression alone, using mutations doesn't discriminate between the subtypes too well, although it does group most of the luminal A subtypes together, albeit with some of the other subtypings as well. So subtyping the tumors this way using mutations isn't the most accurate, but whether these specific subtypes may play a role in treatment susceptibilities is something that we are planning to look at as we get more samples. So this is the variants that we capture in the solid tumor biopsies, and just looking at the same plots, but in the cell-free DNA. So as you can see, firstly, that there are quite a few mutations that are overlapping with both the or genes that are mutated in both the cell-free DNA and the solid tumors. So for example, TP53, KMT2D, etc. We do capture some variants that are mutated in both sample types, but it also does appear that we do capture a lot more recurrently mutated variants in cell-free DNA, and we have done some filtering with VAFs, but the problem is we don't have a matched normal. So as you can see, we do have some mutations that it's not clear whether or not they are specific to the tumor, and using these variants doesn't group any of the subtypes together, and we think that a lot of these recurrently mutated variants are likely playing a noise. So at the moment, to cut down on the number of variants, what we did try to do is try to look at the overlapping variants between the cell-free DNA and the solid tissue biopsies. So one thing is that the gene panel that is being used to profile mutations in both the cell-free DNA and the solid tissue biopsies is slightly different. We do capture a lot more. I think the gene panel for the solid tissue is a lot larger compared to the cell-free DNA one. And looking at this upset plots, what it's essentially showing you is that we have 47 genes that are overlapping between the solid tissue and the cell-free DNA panel. And if we actually try to look at between samples that have both a solid tissue and a cell-free DNA biopsy profile so far, we have about 48 samples. And of those 48 samples, we've found within 38 patients, there are recurrent mutations or concordant mutations between the solid tissues versus the cell-free DNA samples. So what I'm showing you in this plot here on the x-axis is the variant allele frequency of the variants captured in the solid tissue. And on the y-axis is the variant allele frequency of the same mutation, but the VAF in the cell-free DNA. And as you can see, we do capture quite a few variants, particularly in the common drivers such as TP53, KMT2C, etc. But we notice that there are typically two situations or two groups of variants. The first are variants that typically have a low cell-free DNA VAF, but it's a variable VAF in solid tissues or slightly higher. And we suspect that these are most likely the best candidates because typically you would expect the VAF of a tumor-specific mutation in cell-free DNA to be a bit lower. Whereas in this other group of variants, we see that there's almost a perfect correlation in VAF between the solid tumor and ctDNA. And these could partially be driven by copy number changes, but we suspect that some potential germline variants or variants from R chart are sort of creeping in here. So at the moment, while we do detect overlapping variants, we're still trying to identify which would be most informative later on. So far, we've only profiled the baseline cell-free DNA samples, as well as I think most of the solid tissue DNA profiles, but we're still working on a few things with respect to these data sets. The first is to complete the ctDNA targeted sequencing. So we've only profiled the baseline samples so far, and at the moment we're looking to profile samples that were collected after the start of treatment, so at week 12, from the baseline samples, as well as samples that were collected upon detection of recurrence. And once that's done, what we're trying to do here is we're trying to identify specific subtypes that might be susceptible to palbociclib. And whether this is using mutations or transcriptional signatures remains to be seen. We suspect that transcriptional signatures will likely yield the best markers indicative of whether an individual is susceptible to treatments. But the advantage of using mutations in cell-free DNA is that it's minimally invasive. If we look at cell-free DNA, and we can collect repeated samples, unlike solid tissue biopsies. And throughout the course of treatment, we suspect that there'll be changes in clonality of certain clonal subpopulations within the tumor. And this could be potentially better captured using mutations versus a single time points looking at transcriptional signatures. Thirdly, while we do want to look at susceptibility to treatments, we want to use the ctDNA variant allele frequencies or the changes in frequency of the variants in ctDNA to assess whether or not someone is progressing well or is responsive to treatments, but also understand whether or not someone is indicative of having a recurrence in the future. And finally, we also do want to see if we can predict subtypes using cell-free DNA mutation signatures, as I try to show with some of the PC plots. But perhaps there are also other transcriptomic signatures that can be potentially better captured using the cell-free DNA. And to finally use all this data to correlate with clinical outcomes. So I'll turn it back to Jane for this closing slide before we move on to questions.
Great, thank you. And I apologize, I didn't realize my video was off. So thanks, Nick. So as Nick summarized, the ongoing work will focus on key changes and the patterns of change through the longitudinal sampling of these patients defined by their progression status. And we have the rest of the clinical variables collected by the trials to do those clinical correlations. The solid tissue profiling, both genomic and transcriptomic, will help to anchor these longitudinal samples to determine whether resistant clones may have already been initially present or acquired during their treatment. The big picture then is to really develop a rational profiling strategy that adequately captures the information at the time of diagnosis through to the patient journey, so that we're reducing the unnecessary treatment of patients with agents that they may not benefit from. And at the same time, accelerating treatment decision-making when markers of progressions appears. And so with that, I will end and stay tuned for updates as we progress through the rest of the samples. Next slide, Nick. Great, and of course, we couldn't do this without the collaborative efforts from a number of teams here at OICR and our external collaborators. So thank you very much, and we'd be happy to take questions at this time.